Hello, good afternoon. My name is Sean Garrett-Rowe from the University of Pittsburgh. I have the pleasure to be your host for the last session of today's talks. We have a lovely program. Our first announcement that we want to make sure is clear is session number 3 is 4 30-minute talks. So the session will end at 5:30. I was at the half hour. I'm looking at a 3:30 back here, and I'm on East Coast time. So I'm very confused, but it'll be at the 30-minute mark. We have 4 30-minute talks. The first talk for today, there's been a change in the schedule. It'll be Albert Liu, who is able to be here to talk about his PhD research while he was at Michigan. Albert is currently at the Max Planck Institute for Structure and Dynamics of Matter in Hamburg. So Albert, we look forward to your presentation. Please, the floor is yours.
Great. Hello, everybody. So as Sean mentioned, Steve Cundiff couldn't be here today. So I hope I can entertain you just the same. And as Sean also mentioned, I'm currently at the Max Planck Institute, but today I'll talk about some work that I've done in Steve Cundiff's group at the University of Michigan, where we studied colloidal nanocrystals using 2D spectroscopy with a new angle of pushing our measurement temperature regime down to cryogenic temperatures to truly measure the intrinsic properties of these nanocrystals that we're studying. So I think a good place to start is to define what is a colloidal nanocrystal. So colloidal nanocrystals are pieces of material whose dimensions are on the nanometer length scale, and they're colloidal, which means grown in solution. And in these nanocrystals, quantum confinement, due to their small size, gives rise to discrete energy levels. So by changing the nanocrystal size, you can change the energy gap of the material. And this provides a convenient way to tune the colors of light that these nanocrystals absorb and emit at. And this is the main reason why they found so many practical applications. So for example, here's absorption of fluorescence spectra of lead sulfide nanocrystals, where you can see red-shifting absorption of fluorescence with increasing nanocrystal size. What I want to point out, though, is that these peaks just look like broad Gaussians without any obvious structure on them. But if we compare the fluorescence of an ensemble to that of individual nanocrystals, you can see that these broad Gaussians are actually composed of narrower line shapes from individual nanocrystals. And this shows that inhomogeneous broadening dominates the linear optical properties of an ensemble. So this gives you an idea for some of the difficulties with studying the optical properties of these nanocrystals, since in an ensemble, you just get these broad line shapes that only give you information about the size distribution of the ensemble, while the fluorescence spectra of individual nanocrystals varies dramatically even within the same batch of nanocrystals. And if you want to design a device using these nanocrystals, you want to know about their ensemble average properties, since you don't want to design a device around the properties of a single exceptionally good or exceptionally bad nanocrystal. So this is the situation I described on the previous slide, where you have nanocrystals of different sizes, all emitting light at slightly different wavelengths. And we want to know about their ensemble average properties, but their individual spectra all combine to give us this broad and homogeneously broadened spectrum. So how do we unfold this inhomogeneous distribution to get at the underlying homogeneous line shapes? So the most powerful way to do this is to correlate absorption and emission spectra. So in other words, if my sample absorbs light at a certain wavelength, what wavelengths does it emit at? And of course, as everyone in this audience knows, this is exactly what 2D spectroscopy does, where you get a 2D spectrum that looks something like this, where the y-axis is absorption frequency and the x-axis is emission frequency. And in the context of colloidal nanocrystals, each point along this diagonal line where absorption equals emission corresponds to nanocrystals of a certain size, and the line shape in the cross-diagonal direction, or the perpendicular direction, reflects its homogeneous properties. So of course, 2D spectroscopy of colloidal nanocrystals isn't a new idea and has been done by many different groups around the world to reveal all sorts of interesting physics. But almost all of these studies have been performed at room temperature. And the issue with room temperature spectra is that peaks are severely affected by thermal line broadening, to the point where homogeneous line widths are often comparable to the inhomogeneous line widths. If you lower the temperature, however, intricate line shapes appear that give you access to a whole new range of physics. So for example, there was this beautiful work that came out of the groups of tonopolarites and Gonata zygmontis, where they observed narrow exton line shapes at liquid nitrogen temperatures. So this was our motivation to try a 2D spectroscopy of colloidal nanocrystals at even lower liquid helium temperatures to really try to measure the intrinsic properties of extons of these nanocrystals. So in my talk, I'll focus on a specific new class of nanocrystals called perovskite, cesium-lead halide perovskite nanocrystals, which have a chemical formula cesium-lead X3, where X is a halide atom, chlorine, bromine, or iodine. And this material crystallizes in a perovskite lattice, hence the name. And these nanocrystals can be grown as either cubes or platelets with dimensions on the nanometer length scale. And these perovskite nanocrystals have attracted tremendous attention in the nanocrystal community in recent years due to their unique optical electronic properties. So for example, one can obtain quantum yields near unity from a bare perovskite nanocrystal without additional synthesis complications such as alloying or growth of additional shell layers on top. There's also an additional tuning knob for the emission color, which is the halide atom X. And as you can see in the figure at the bottom right, one can tune the emission color all the way from purple to deep red by changing the halide atom without changing the quantum confinement. So in my talk today, I'll talk about cesium-lead iodide perovskite nanocrystals in two different nanocrystal geometries. So first, I'll discuss exton fine structure in perovskite nanocubes and how we use 2D spectroscopy to resolve interband and intraband quantum coherence of the fine structure states. I'll then move on to talking about perovskite nanoplatelets, where the change in quantum confinement dramatically reduces disorder of the exton resonance. And I'll present how we use 2D spectroscopy to measure their intrinsic line widths as well as their line broadening mechanisms. And with that, we'll start with fine structure in perovskite nanocubes. So the exton fine structure in perovskite nanocrystals is one of their most discussed properties. And it all started with this Nature paper in 2018, which presented evidence that the exton band edge in these perovskite nanocrystals is composed of a dark state and 3 non-degenerate bright states. And what's so special here is that the dark state is actually highest in energy, whereas in other materials, the dark state is always lowest in energy due to the electron-hole exchange interaction. And this is because in perovskite nanocrystals, there's an additional Rashba spin-orbit interaction that both lists the degeneracy of the triplet states as well as lowers them in energy. And this was quite exciting for the whole community since this meant that extons being generated in these perovskite nanocrystals could directly emit light instead of having to first relax into an intermediate dark state with a much longer radiative recombination time. But in 2019, another paper came out that studied these perovskite nanocrystals using magneto-PL spectroscopy, which presented pretty convincing evidence that the lowest exton state in their system was actually dark. And this caused quite a controversy in the community and was one of the motivations for us to apply 2D spectroscopy to study the exton fine structure in these nanocrystals. So one of the most useful properties of these bright triplet states that I haven't mentioned yet is that their dipole moments point in orthogonal directions. So to measure quantum pathways involving these triplet states, we vary the polarization of our second excitation pulse, which in this diagram is pulse B. And below the pulse diagram, you can see the relative polarizations of each excitation pulse in the laboratory frame. And we call the first polarization scheme collinear polarization and the second one cross-linear polarization. And this is just a quick overview of our experimental setup. Our excitation pulses are focused onto our sample in the boxcar geometry, which results in a photon echo Fourier-Vixen signal being emitted in a unique direction. Our excitation pulses are generated by an OPA, tunable from roughly 570 to 660 nanometers at a repetition rate of 250 kilohertz. And the pulse duration varies a bit with the excitation wavelength, but is roughly 100 femtoseconds or below. And the perovskite nanocubes that we study have an average side length of roughly 8.7 nanometers, which for comparison, the exton bore diameter of bulk cesium lead iodide is 12 nanometers. And to study these nanocrystals at cryogenic temperatures, we suspend them in a liquid called heptamethylonate, which is a long-branched alkane that forms a transparent glass at temperatures below 100 Kelvin. So this is what the 2D spectra look like for collinear and crosslinear polarization. There's a lot going on here, but here we're just interested in what changes between the different polarizations, since this is what has to do with the triplet states. So to take a closer look, we can take cross-diagonal slices along this red dashed line, and we can see that the main differences are these peaks in the middle, which correspond to absorption and emission in and out of different triplet states. So let's start with the collinear spectrum. So peak 2 in the middle lies on the diagonal. So this corresponds to absorption and emission in and out of the same triplet state. It's then suppressed in the crosslinear spectrum due to the orthogonal dipole moments of the triplet states. So peaks 1 and 3 lie at plus and minus omega 2. So they correspond to coherent coupling between states X and Y in the level diagram on the right. And peaks 4 and 5 lie at plus and minus omega 1. So they correspond to coherent coupling between states Y and Z. So one of the odd features of these slices is that peaks 4 and 5 are much broader than the others. So in fact, when we fit these peaks to extract the dephasing rates of these triplet state coherences, we found that states X and Y shared relatively similar dephasing rates, while state Z dephased much more rapidly than the others. So our hypothesis for why this was the case is that in cesium lead iodine nanocrystals, the dark state actually lies between states Y and Z. And this dramatically increases the dephasing rate of state Z by introducing an additional fast non-radiative relaxation pathway. And this is perfectly corroborated by single nanocrystal experiments of nearly identical samples, where they observed polarized doublets separated by omega 2, but never a third peak separated by an energy omega 1. So because we see signatures of optical triplet state coherences in our 2D spectra, we should in principle be able to generate as well as measure coherences between the triplet states as well. So these intertriplet coherences should show up as sidebands in what are called zero quantum spectra, which we measure by scanning the intermediate waiting time, capital T. So we first tried measuring zero quantum spectra with collinear excitation, but we didn't see any clear intertriplet coherences between states X and Y. This may be because the energy splitting was too small compared to the intraband dephasing rates, but we really don't know. We did, however, see many signatures of vibrational coherences, so I have some data on that in the extra slides if there's interest. This is what the zero quantum spectra look like for crosslinear excitation, where we see two clear sidebands that correspond to coherences between states Y and Z. And if we take slices along this red dashed line as a function of temperature, which is plotted in the top right, we see that these intertriplet coherences are quite robust against thermal dephasing, since the line width doesn't change as we increase the temperature up to 40 Kelvin. And if we fit a slice, say, at 20 Kelvin, we can extract an intertriplet coherence time of 1.36 picoseconds. And to motivate a possible application of these intertriplet coherences, I'd like to draw an analogy to valleytronics in 2D materials, where circularly polarized valleys in the band structure are used for quantum information. And even though the dephasing times in these 2D materials are quite short, it was actually shown that you could actually transfer exciton polarization from one valley to another with a terahertz pulse within a single optical cycle. So one could imagine performing similar operations on a perovskite nanocrystal with linear instead of circularly polarized initialization and readout. And if we compare the inter-valley coherence time of, say, tungsten diselenide of 98 femtoseconds to the intertriplet coherence time of 1.36 picoseconds, our intertriplet coherence time compares quite very favorably. So now I'll switch gears to another material that's still composed of cesium lead iodide, but in a different nanocrystal geometry. And just like when going from quantum dots to quantum wells in conventional semiconductors, the change in quantum confinement dramatically modifies the electronic properties of the excitons. So before jumping into to any 2D spectra, I think it's actually useful to take a step back and look at absorption spectra of the two nanocrystal geometries, since one can actually observe dramatic differences even in ensemble spectra. So here you can see absorption spectra of the perovskite nanocubes as a function of temperature, where we see a distinct peak corresponding to the lowest exciton transition. And we can fit this peak to pull out a center energy and line width as a function of temperature. And if we look at the dependence of the center energy on temperature, we find that the exciton resonance blue shifts with increasing temperature due to exciton phonon coupling. And the temperature trend has this funny curvy shape because excitons in these nanocubes couple not only to acoustic phonon modes, but also multiple optical phonon modes of the cesium lead iodide at lattice. The peak width, on the other hand, doesn't change very much with temperature, which suggests that the peak width is simply the inhomogeneous line width of the ensemble. If we look at comparable absorption spectra of the perovskite nanoplatelets, we immediately see that the absorption line widths are much narrower. And in fact, the energy windows of these two plots are the same, so you can directly compare the peaks by eye. And if we look at the fitted center energies as a function of temperature, we find that the center energies vary linearly with temperature, indicating that exciton phonon coupling is primarily with acoustic phonon modes. And there's actually more action with the peak widths, which decrease with temperature until saturating at around 30 or 40 Kelvin. So this indicates that the absorption line widths at least partially reflect the actual exciton line shapes rather than just the inhomogeneous line width of the ensemble. So why is the disorder so different between these two nanocrystal geometries? So in nanocubes, excitons that you generate experience three-dimensional quantum confinement. So any variation in the nanocrystal shape or size is translated into a distribution of resonance energies. And the usual case where this resonance energy distribution is much larger than the line shape of a single nanocrystal is shown in the top right. And we call this line shape inhomogeneously broadened. In nanoplatelets, the situation is quite different. So in nanoplatelets, excitons experience dominant one-dimensional quantum confinement. And because the thickness of these nanoplatelets can be controlled very accurately down to single unit cells, this dramatically reduces inhomogeneous broadening to the point where it's often claimed that spectral line shapes in these nanoplatelets are homogeneously broadened. And this is a claim that has implications for a variety of practical applications, ranging from lasing to transport and superlattices, and is usually justified using plots like the one in the bottom right, where the photoluminescence of an ensemble is compared to that of a single nanoplatelet and is shown to exhibit similar widths. We were a bit skeptical of this common assumption, however, since, as you can see in the top right, the single particle line shapes can vary really dramatically even within an ensemble. So when you perform these single nanocrystal measurements, you don't know whether you're looking at a representative nanocrystal or just an outlier. So to truly claim homogeneous or inhomogeneous broadening of your ensemble, you really need to perform measurements of an ensemble. So in these nanoplatelets, there are basically two dimensions we have to consider now, which are the lateral dimensions of our nanoplatelets, as well as their thicknesses. So if you look at the absorption spectra of our samples on the right, you can see two distinct exciton absorption peaks corresponding to two different nanoplatelet thicknesses. And these thicknesses of 2-layer and 3 layers refer to the tetrahedra formed by the lead and iodide atoms, which for reference is around 6 or 7 angstroms long. And the 2-layer nanoplatelet absorption peak is higher in energy due to the tighter quantum confinement of the excitons. And for reference, the lateral dimensions of our nanoplatelets are around 10 nanometers, which is quite large compared to the exciton bore diameter of cesium lead iodide in 2D of 6 nanometers. So it's clear that the dominant quantum confinement is from the outer plane thickness. However, variations in the lateral confinement can still introduce inhomogeneous broadening of your exciton resonance. So this is what the 2D spectra look like for our 3-layer nanoplatelets at temperatures of 10 and 60 Kelvin. where we observe textbook inhomogeneously broadened resonances. And as a reminder, the line shape in the elongated direction reflect the inhomogeneous line width, while the line shape in the cross-diagonal direction reflects the homogeneous line width at a given resonance energy. And for comparison, here are 2D spectra of the 2-layer nanoplatelets where we observe dramatically reduced homogeneous and inhomogeneous line widths. And to measure the homogeneous line widths, we can take slices along this dashed white arrow. And in these plots on the right, the data is shown as the shaded curves, while corresponding fits are shown as these dotted red lines. And I'll make these statements more quantitative on the next few slides. So as a reminder, the functional form of the cross-diagonal line shapes is shown here as the square root of Lorentzian. And as a disclaimer, this is the limiting form for dominant inhomogeneous broadening. But in our actual fits, we use a more general functional form for arbitrary inhomogeneity. And the parameter gamma is the homogeneous line width that we want to extract. And this contains 3 main contributions. So there's first the intrinsic line width gamma naught, which is one can think of as the natural line width of an isolated oscillator at 0 temperature. And this is essentially limited by population relaxation. The second contribution is a linear dependence on temperature. So this is due to exton-phonon coupling with acoustic phonon modes. And the last contribution is a dependence on exton density. So this reflects broadening due to exton-exton scattering. So to extract the thermal broadening parameter A, we took measurements of the homogeneous line width as a function of temperature, which unsurprisingly exhibit a linear dependence on temperature due to the predominant coupling to acoustic phonon modes. And linear fits to the data are shown as the dashed red lines, as well as the equations on top, which show that the thinner 2-layer nanoplatelets exhibit both a narrower 0 temperature line width of 0.45 MeV compared to 1.67 MeV, as well as the reduced thermal broadening parameter of 0.032 MeV per Kelvin versus 0.045 MeV per Kelvin for the 3-layer nanoplatelets. And if we extrapolate this homogeneous line width to room temperature and compare it with the inhomogeneous line width, we can reach a concrete answer to the question I posed at the beginning of this part of my talk, which was, are perovskite nanoplatelets homogeneously broadened? And the answer, it turns out, is it depends. So for the 2-layer nanoplatelets, the extrapolated homogeneous line width is comparable to the inhomogeneous line width sigma at room temperature. Whereas for the 3-layer nanoplatelets, there's still a significant difference at room temperature. So this means that the common assumption of homogeneously broadened exton resonances at room temperature should be applied with care in thicker colloidal nanoplatelets. And we also performed measurements as a function of excitation density. And to account for differences in optical absorption, we converted the excitation fluence to extons per square centimeter as the x-axis. And the fitted broadening parameter B, which is the slope of these linear fits, we also find to be smaller for the thinner 2-layer nanoplatelets, which indicates that they're also more robust against excitation-induced dephasing. So to summarize this part of my talk, we applied 2D spectroscopy at cryogenic temperatures to colloidal nanoplatelets to measure their intrinsic line widths and line broadening mechanisms. And we find that decreasing nanoplatelet thickness comes with a host of beneficial effects. So for example, decreased homogeneous and inhomogeneous line widths, decreased exton-phonon coupling, as well as decreased excitation-induced dephasing. And I should also mention that the results that I've presented today are only part of the full study that are published at this reference at the top left, where we performed further analysis of the influence of lateral confinement on the broadening mechanisms. So with that, I reached the end of my talk today. So I'd like to thank Department of Energy for funding this research. I'd also like to thank Professors Lázaro Padilla and Nano Noguera for providing the perovskite nanocrystal samples, and of course, Professor Steve Cundiff for supervising this research. And the results that I've shown today are published in these 2 references shown here, and I'd be happy to take any questions.
Thank you. Thank you very much for the lovely talk. The floor is open for questions. And so we have one question here is first. I'm sorry, you'll be second. Please.
Should I go?
Ahead. Yes.
Okay. Thank you. It's a beautiful talk, and it's always a pleasure to see Steve's work, and you've done great work here. There's something that you might be familiar with, and the audience might not be, because you've looked at perovskites. You're looking at the temperature dependence. At 4 Kelvin, the system is not in the same state that it's in at 300 Kelvin, because at 300 Kelvin, it's glassy and disordered. And at 4 Kelvin, you freeze out that disorder. So moreover, at 4 Kelvin, it seems to be that you have weaker electron-phonon coupling, and you also have delocalization of the exciton, which gives rise to the fast, the so-called giant oscillator strength. So there are all these effects that happen at 4 Kelvin that don't seem to be happening at 300 Kelvin. We've not done the low temp experiment. So my question for you is, what differences do you see at 4 Kelvin than, let's say, at your higher temperature, 50 or 60 Kelvin, that might suggest that the electronic structure is very different? And the other question is, as you pointed out to the Nature and the Nature methods or Nature materials paper, there's the controversy of the fine structure. Have you been able to gather insights onto that fine structure from your data here?
So starting with your second question. So in fact, this fine structure at the time when we looked at this, there was quite a lot of controversy. People were reporting different level orderings for different types of perovskite nanocrystals. And then we were the first to look at the cesium lead iodide fine structure. And we saw something that suggested that there was this interleaved level ordering. And in fact, after we reported this, there was actually a theoretical paper that came out by Peter Sersel that looked at basically the influence of nanocrystal size and anisotropy on the level ordering. And it turns out that this is a very finely tuned effect, because there's this competition between Rashba spin-orbit coupling versus the electron-hole exchange interaction. So we were in this sort of weird region where there was a crossover between the level orderings. And in fact, if you look at the slice dependence, which is in the supplemental info of the paper, you see that actually the dephasing rates of the lower two triplet states, X and Y, actually start increasing quite dramatically. And we think that this might be where the nanocrystal size is experiencing this crossover between the level ordering. And as for your first question, we don't actually have very many measurements at high temperatures. So we have a very narrow bandwidth system. Well, I say have, we had. Now Steve has some fancy new lasers, but at the time we had very narrow bandwidth lasers, and we couldn't get very good room temperature spectra. And if you look at the temperature dependence of these one quantum spectra of, say, the nanocubes, at around 15 Kelvin, the fine structure is completely washed out. So we don't see any dramatic, let's say, dramatic changes that would indicate like a structural phase transition or a disorder transition. That
what about the phonons, for example? There's a whole mystery of whether you should see them or not, because if they're glassy or non-glassy. I mean, so in your zero quantum, you don't see any phonons. All you see is fine structure.
No, we see vibrational coherences.
You do, okay.
So these are the zero quantum spectra for collinear excitation, where we see side bands due to vibrational coherences. And one thing that indicates that they're vibrational is the asymmetry of the side bands. Whereas in the electronic side bands, you see that they're symmetric around the zero energy line. And we actually see, if you look at the zero quantum spectra, they're actually as a function of the first time delay tau. And we see that the sidebands only come in after a certain time delay tau, which might indicate polaronic dynamics. And in fact, if we look at the zero quantum spectra as a function of temperature, we also don't see some dramatic crossover. So this is a very interesting topic, but I don't actually have any data that indicates any of this.
Yeah. Brief question. Okay, I had two, but okay, a very brief technical question about this polarization measurement. So you actually showed only 3 polarizations, but what is the detection polarization what you're using?
So we put an orthogonal polarizer before a spectrometer. So we're filtering out only
So it's
made it-
XY, XY, right?
XY, XY. Yes.
Okay, thank you.
Yeah.
Thank you very much.